Hello everyone, myself Dr. Amanpreet Kaur Kalsi. In this presentation, we'll take up another topic of lipids. The topics from lipids that we will cover in this presentation are Introduction to lipids, fatty acids, classification of fatty acids, nomenclature of fatty acids, classification of lipids, amphipathic lipids, and biological importance of lipids. Let's start with Introduction to lipids. Lipids are a heterogeneous group of compounds which includes fats, oils, steroids, waxes, etc. These lipids are related more by their physical properties than the chemical properties. Which are those physical properties? First, that these are relatively insoluble in water, and second, that they are soluble in non-polar solvents such as ether and chloroform. Talking about fatty acids contain mainly non-polar carbon-carbon and carbon-hydrogen bonds. As shown over here, the black-colored balls are carbons and the white-colored balls are hydrogens. So there are many carbon-carbon, carbon-hydrogen bonds which are all non-polar, that means hydrophobic. And then there are few polar bonds of carbon oxygen and hydrogen, which are hydrophilic. Shown above is the skeletal structure of a fatty acid, and still above that is an example of fatty acid, palmitic acid, which is a 16 carbon fatty acid. Next, coming to the classification of fatty acids. Fatty acids can be classified into two broad categories, saturated and unsaturated. Saturated fatty acids, they have single bond between all the carbons, and because of single bonds they have this kind of structure and thus can be tightly packed together. Unsaturated fatty acids have one or more double bonds between the carbon atoms. Because of the double bond nature, they have kinks or bends, and because of this in saturated fatty acids, because of this closed packing, they are mostly solid at room temperature, and the same reason, because of the kinks or bends of unsaturation of double bonds in the unsaturated fatty acids, they are mainly liquid at room temperature. Saturated fatty acids are derived from animal sources except coconut oil. That means ghee, butter, etc. They are all saturated fatty acids. And unsaturated fatty acids are derived from plant sources like olive oil, mustard oil, etc. Saturated fatty acids are not susceptible to further oxidation because there are no double bonds available, and unsaturated fatty acids are susceptible to further oxidation when exposed to atmosphere. High amount of saturated fatty acids can be harmful to health. On the contrary, unsaturated fatty acids are associated with various health benefits. Some examples of saturated fatty acids are 4-carbon butyric acid, 16-carbon palmitic acid, 18-carbon stearic acid, etc. And if we talk about unsaturated fatty acids, they can be further classified into three categories: monounsaturated, polyunsaturated, and eicosanoids. Monounsaturated fatty acids, as the name indicates, they have single double bond of unsaturation, and polyunsaturated fatty acids have more than one double bonds. Both are liquid at room temperature. Monounsaturated fatty acids have lower melting point compared to saturated fatty acids, and polyunsaturated have still lower melting point. Both monounsaturated and polyunsaturated fatty acids help in reducing the risk of heart diseases. So these are good fatty acids. Next is showing one example of each. 18-carbon oleic acid is an example of monounsaturated fatty acid, and 18-carbon linoleic acid is an example of polyunsaturated fatty acid, which has two double bonds in its structure. Some fats containing monounsaturated fatty acids are olive oil, canola oil, peanut oil. Likewise, some fats having polyunsaturated fatty acid molecules are soybean oil, corn oil, and fatty fish. The third category of unsaturated fatty acids was eicosanoids. Eicosanoid term is derived from eicosa, which means 20 carbon. So all the fatty acids which have 20 carbons or are derived from 20 carbon molecules are called eicosanoids. Examples are prostanoids, leukotrienes, and lipoxins. Shown below are further examples of eicosanoids. Arachidonic acid, which is a 20 carbon fatty acid, is an example of eicosanoid. Prostaglandin, this is the structure of prostaglandin F2. This is again an eicosanoid. It is derived from 20 carbons. And leukotrienes. Prostaglandins have very important role to play in our body. They occur in every mammalian tissue acting as local hormones. They are synthesized in vivo by cyclization of center of carbon chain of the 20 carbon eicosanoid molecule. So if this is a 20 carbon molecule, the central carbon, carbon number 9, 10, 11, undergoes cyclization, and then the structure is called prostaglandin. Thromboxanes, these are cyclopentane ring interrupted with an oxygen atom. So if this is a prostaglandin, this cyclopentane ring, if it is interrupted with an oxygen molecule, this ring is now called an oxane ring, and the molecule is now called a thromboxane molecule. Leukotrienes and lipoxins, these are a group of eicosanoid derivatives formed via lipoxygenase pathway. These are characterized by the presence of 3 or 4 conjugated double bonds respectively. They cause bronchoconstriction. These are potent pro-inflammatory agents and play a role in asthma. Shown in this figure is a leukotriene B4 molecule. You can see 3 conjugated double bonds in its structure. Next, coming to the nomenclature of fatty acids. If it is a saturated fatty acid, it is named with parent hydrocarbon plus enoic. Example, 8-carbon fatty acid is named octanoic acid. If it is an unsaturated fatty acid, then prefix enoic is added. Example, 18-carbon unsaturated fatty acid is named as octadecanoic. Octadec for the 18 carbon and enoic for the unsaturation. The carbon atoms are numbered from carboxyl carbon, numbering as carbon number 1 as shown over here, and then rest of the carbons are numbered 2, 3, 4, 5 and so on. Carbon atom adjacent to carboxyl carbon, that is carbon number 2, 3 and 4, they are also named as alpha, beta, gamma, delta and so on. The other way of numbering is starting with the methyl carbon, that is opposite the carboxyl one. Terminal methyl carbon is also known as omega carbon or N carbon, and then starting from the methyl carbon walking towards the carboxyl carbon, these are then numbered omega 1, omega 2, omega 3 and so on. Various conventions also use delta for indicating the number and position of double bonds. For example, delta 9 indicates a double bond between carbon 9 and 10 of fatty acid. So 18:1 semicolon 9 means fatty acid with 18 carbons having one double bond at carbon number 9. That means between carbon 9 and 10. This can also be written as delta 9 18 colon 1. Omega 9 indicates a double bond on ninth carbon counting from the omega side. So if we write omega 9 C18 colon 1, it means now the double bond is between carbon number 9 and 10 starting from the methyl end. Next, coming to lipids. Lipids can broadly be classified into three categories: simple lipids, complex lipids, and precursor or derived lipids. First, coming to simple lipids, these are esters of fatty acids with various alcohols and can be of two types, fats and waxes. We'll take up fats first. Fats are the esters of fatty acid with glycerol. So when the alcohol is only glycerol, it is called fat. How are these ester bonds formed? Let's see in detail. The fatty acid molecule we have just studied. These are chains of carbon atoms with methyl group at one end and carboxylate group at the other end. So this is a fatty acid molecule. And glycerol forms the backbone of lipids. It is a water-soluble alcohol. This is the structural formula of glycerol: 3 carbons and 3 hydroxyl groups. So when ester formation occurs, the interaction occurs between the glycerol, one molecule of glycerol, and 3 molecules of fatty acids. This is one molecule, this is second molecule, and this is the third molecule. What happens? OH from one fatty acid and hydrogen from the glycerol is released, giving rise to 3 molecules of water. Here occurs the condensation reaction and the product formed is called triacyl glycerol. Glycerol attached with 3 molecules of fatty acids. And in this, 3 ester groups are formed, which are labeled in red. This is the block diagram of a simple lipid, that is fat, glycerol forming the backbone, and 3 fatty acids attached to glycerol via ester bonds. These triacylglycerols undergo metabolism to form glycerol and 3 fatty acids, just the way it was formed. The fats are stored in the body in the form of adipose mass. It is stored in adipose cells, and when energy is required, these triacylglycerol undergo metabolism, forming carbon dioxide, water, and energy is released. Second category of simple lipids are waxes. These are esters of fatty acids with higher molecular weight monohydric alcohols. So over here, alcohol is higher molecular weight and not only glycerol. This is a general structure of wax. So in between is the ester bond. There is a long chain of alcohol and long chain of fatty acid. How it is formed, how waxes are formed: fatty acid molecule, alcohol molecule, esterification occurs between the red part of fatty acid and the blue part of alcohol shown over here to form the wax molecule, and again water molecule is released. This shows the fatty acid section of a wax. This shows the alcohol section of the wax. So both the parts, fatty acid as well as alcohol parts in waxes, are very long and complex, which are joined together by ester bond. After simple lipids, the second category of lipids is complex lipids, which are of 3 types: phospholipids, glycolipids, and other complex lipids. Complex lipids are esters of fatty acids containing groups in addition to alcohol and fatty acid. So the name itself indicates these are complex. So in addition to alcohol and fatty acid, they have some other compound as well. Which are those compounds? Phospholipids, glycolipids, and rest of the complex lipids. Let's talk about phospholipids first. Phospholipids are lipids containing, in addition to fatty acid and an alcohol, a phosphoric acid residue. This is the phosphoric acid residue which is present in the lipid molecule along with fatty acid and alcohol, and these are attached by phosphodiester bond. So if this is a simple lipid, triacylglycerol, here comes the phosphorus group and now it becomes phosphoacylglycerol. Sphingomyelin is another example of phospholipid. These phospholipids frequently have nitrogen containing bases and other substituents also. Example in sphingosine, you can see a nitrogen molecule is also present. Over here, the detailed structure of sphingomyelin. Here is the nitrogen molecule, here is the phosphorus molecule. And this is the block diagram of sphingomyelin. These phospholipids, we all know, form the lipid bilayer of all the cells in living organisms because of their non-polar tail and polar head. The second kind of complex lipids are glycolipids. As the name itself indicates, they have glycogen or carbohydrate molecule along with fatty acid and alcohol. Over here is shown an example of glycolipid where this is the fatty acid chain, this is the sphingosine molecule, and here is the carbohydrate unit. Also shown over here. So this is the fatty acid part, this is the alcohol part, and this is the carbohydrate molecule attached. The third category of complex lipids is other complex lipids, which include sulfolipids, aminolipids, lipoproteins, etc. Here is shown an example of sulfolipid where you can see a molecule of sulfated glucose attached to the fatty acid residue. The third category of lipids are precursor and derived lipids. These include fatty acids, glycerol, steroids, other alcohols, fatty aldehydes, ketone bodies, hydrocarbons, lipid-soluble vitamins, and hormones. So all these come under precursors and derived lipids. A very important precursor and derived lipid is steroid. The basic skeleton of steroid is shown here. It has 4 rings, out of which 3 rings A, B and C are 6-carbon rings, and the 4th ring is a cyclopentane ring. Along with it, there are 2 methyl groups also present. This is the basic steroid skeleton. Numbering of steroid skeleton is very specific. It is numbered starting from this carbon numbered 1, then 2, 3, 4, 5, 6, 7, 8, 9, 10. Here is the 11th carbon, 12th, 13, 14, 15, 16, 17. Then the methyl carbon attached with carbon 13 is numbered carbon 18, and another methyl group attached at carbon 10 is numbered carbon 19. The best known and most abundant steroid in body is cholesterol. Cholesterol is formed in brain tissue, nerve tissue, and bloodstream. It is the major compound found in gallstones and bile salts. And here is the structure of cholesterol. In addition to the 4-ring structure, there is present elongated side chain at carbon 17. And total carbons in the cholesterol are 27. Next comes amphipathic lipids. Lipids where part of the molecule is hydrophobic or water insoluble and part is hydrophilic or water soluble are described as amphipathic lipids. As you can see in the figure also, there is a long non-polar or hydrophobic group and a small polar or hydrophilic group. Examples we now know are fatty acids, phospholipids, sphingolipids, bile salts, and to a lesser extent, Cholesterol also is an amphipathic molecule. These amphipathic lipids become oriented at oil-water interfaces with the polar group in the water phase and the non-polar group in the oil phase. A bilayer of such amphipathic lipids is regarded as the basic structure in biologic membranes. As we can see in the figure, the lipid bilayer forms the cell membrane of all the cells in mammalian body. Amphipathic lipids also form micelles. When they form? When a critical concentration of these lipids is present in an aqueous medium, they form micelles. Micelles are important in facilitating absorption of lipids from the intestine. As you can see in the figure, in the micelles, the lipid molecule arrange in a way that the polar part is towards the aqueous phase and the non-polar part is towards the oil phase. Liposomes are another type of molecules or structures formed by amphipathic lipids. These consist of spheres of lipid bilayers, which enclose part of the aqueous medium, as you can see in the figure also. They are of potential clinical use, particularly when combined with tissue-specific antibodies. They work as carriers of drugs in circulation targeted to specific organs, such as in cancer therapy. When there is a single layer of lipid bilayer, it is called unilaminar liposome. When there are multiple layers of lipid bilayers present, it is called multilamellar liposome. Next structure formed by amphipathic lipids are emulsions. These are much larger particles formed usually by nonpolar lipids in aqueous medium. Amphipathic lipids form a surface layer separating the main bulk of nonpolar material from the aqueous phase. Lastly, let's come to the biological importance of lipids. Lipids are important dietary constituent because of their high energy value. They also have fat-soluble vitamins, essential fatty acids contained in fat of natural foods. The fat stored in adipose tissue, it serves as thermal insulator in subcutaneous tissues and around certain organs. Food stored in our body is mainly in the form of fats. Cholesterol, a steroid lipid, helps in synthesis of vitamin. Cholesterol form hormones like testosterone, estrogen, progesterone, cortisone, cortisol, and aldosterone, which are all important for reproductive, carbohydrate, and mineral metabolisms of our body, respectively. Lipids, as we have seen, as phospholipids, are found in plasma membranes and other organelle membranes of all the cells. Fats provide slow and long-lasting energy. Prostaglandin, which is a derived lipid, helps in induction of labor pain for normal birth of child. Sphingolipids are found in myelin sheath of nerve cells. Glycolipids are found in gray matter and white matter of our brain. So all in all, lipids play a major role in almost all the parts and all the systems of our body. This is all about this lecture. Major parts of this lecture are prepared from the book Harper's Illustrated Biochemistry. Thank you so much.